Assalamualaikum warahmatullahi wabarakatuh. My name is Nurin Izzaty binti mohamad nadzri. My ID student is 2023393257. I'm from class 80-2040, and on this video I will discuss about genetically modified potatoes, which is Solanum tuberosum, for improve the stress resistance and the food safety. Potato is the one of the most important crop in the world. However, its production and nutrient content are threatened by the both biotic and abiotic stresses. Large yield losses are caused by the pests such as the Colorado potato beetle and aphid and viral disease that also significantly affect production. Not only that, the health and safety of the human and animal can also be affected and must be taken. Therefore, the use of genetic engineering can be an efficient tool harmless to the environment and beneficial to the consumer. However, gene transfer has positive effect where it can reduce economic losses, pesticide use, and improve the nutritional properties of the potatoes. The first point is effectiveness of GM potatoes against the pests. Potatoes are modified to be resistant to the pest attack where an alternative occurs related to the CBB, pathogen bacillus thuringiensis, which is BT. BT is a gram-positive soil bacterium that known primarily for the production of the proteinaceous insecticidal endotoxin. There are two groups of insecticidal crystal toxin, namely cry and side protein. This toxin is acquired by the multigenic screen of the side gene family and enter the paraspora inclusion during the sporulation. Cry protein are toxin to the insect of the Lepidoptera, Diptera, Hymenoptera, and Coleoptera, while the side protein are only active against the Diptera and therefore are not widely used. Protoxin dissolved in the alkaline condition, which is activation is done by the proteins such as a trypsin and the chymotrypsin that from toxin fragment. The fragments are able to interact specifically with migate cell receptor and create the pars in the epithelium. This toxin is combination with BT spores is used as a biological insecticide. The main advantage is the specificity of the toxin. As a result, transgenic potatoes containing the Cry3A toxin encoded by the Cry3A gene have been shown to have specific resistance to CPB. About 30 cases of potatoes with integrated Cry3A gene have been approved because these GM plants are able to produce the Cry toxin evenly throughout the plant as a result of the cauliflower mosaic virus constitutive non-specific tissue promoter. Genetic modification of the potato solves the main problem that the protein is produced in the plant and thus protected from the UV. Potatoes are also modified for the safety of human and animal. This is because potato contain a high amount of acrylamide that give a negative effect to the consumer, such as the nausea and the vomiting. To reduce the precursor of the acrylamide formation, a gene silencing cassette was introduced to reduce the expression of the asparagine synthesis 1, and another cassette reduced the expression of a glucan, water dekinase or starch-related R1 gene, and phosphorylase PHL gene. This gene combination significantly reduced acrylamide level in the French fries. Not only that, the inserted gene minimized the amylose content by decrease the level of the granule-bound starch synthesis. This enzyme is responsible for the synthesis of the amylose in the many plants. New efficient genome editing tools, CRISPR or Cas9, are also used to make genetic transfer faster and more efficient than conventional breeding and agrobacterium-mediated transformation. As a result of this modification, the reduction in this number of the black spot is achieved by silencing the polyphenol oxidase 5 gene. They assisted approval GM potatoes with a gene inserted to reduce the asparagine production, reducing sugar level, and the black spot formation. In conclusion, many potato strains are dealt with illegally through genetic modification, and transgenic plants can produce the less metabolites that are harmful to the consumer. Although the use of GM crop is the fastest growing agricultural technology in the world, the biggest drawback of their promising application remains the legal limit. However, it is expected that the legal limit may soon be breached as a result of the current food crisis in the world. The modification method must be done well if the main purpose is to maintain the safety and the health of the consumer using the food and the product. produced, because every modify will have a good bad effect in the long run.